Guys, before we start, I would like to check whether are you already able to see the recordings on YouTube, if the link is working properly. Have you checked that during weekend? Yes, is it working on your side? Perfect. Okay, that's great. So the recordings are, I think, all of. I will check, but I think there should be all of the recordings from both first weeks, and I will then time to time update the channel and upload the other recordings also from this week and the following weeks. Okay. Um, let me let me introduce you with the structure today. Again, as you are already familiar, we will start with some introduction activities, and then we will move to the core. Our main topic for today is supposed to be meiosis. We will see how quick we will be, and then when we are done with meiosis, we can continue with genetics. Um, Yes, so that's everything from my introduction to the session. Do you have any questions, guys, before we start? No? Okay. So everything is in the Google Classroom. Also, recordings are already uploaded, so you can watch them whenever you want. The videos from for the sessions, uh, please watch them when you have time. I think it will be beneficial for you also to understand the complex topics we are dealing with. Okay, so. Let's start with the introduction. Mm, Himari, please, can you read?
Yes. What defines a eukaryotic cell? A. Presence of a cell membrane. B. Presence of a nucleus and membrane-bound organelles. C. Smaller size compared to prokaryotic cells. D. Lack of genetic material.
Thank you very much, Himari. Let's have a few more seconds. Now, let's do a poll. Okay, here are the results. Okay, Himari, let's be quick here. Why have you chosen B?
Because eukaryotic cells have a nucleus and membrane-bound organelles.
Yes, perfect. Why A is not correct, guys? What's wrong with A?
Because both eukaryotic and prokaryotic cell have cell membrane.
Perfect, Ayane. Thank you very much. What about C?
Eukaryotic cells are larger than prokaryotic ones.
Great, Tomohiro. That's perfect. And what about D?
Eukaryotic cells have genetic material.
Yes, exactly, in form of DNA and RNA, and it's mostly concentrated in the nucleus, where also DNA or where the DNA is only place, as well as mitochondria to be found regarding DNA and genetic material. Great, thank you very much, Hitomi. Let's move further. Tomohiro, can you read, please?
What is DNA? A. A type of protein found in cells. B. The energy currency of the cell. C. A molecule that stores genetic information. D. A membrane-bound organelle.
Thank you very much, Tomohiro. Let's have a few more seconds to read it one more time. Okay, here are the results. So, Tomohiro, why have you chosen C?
Um, because DNA contains genetic information inside themselves.
Mm-hmm. Perfect. Okay, so molecule that stores genetic information. Great. What's wrong with with D, a membrane-bound organelle? What membrane-bound organelles do we know already, group? Do you remember? What organelles have their own membrane and they are part of endomembrane system within the eukaryotic cell? Let me show you the picture. It's already almost 2 weeks back. What organelles have their own membrane and are part of endomembrane system? If you remember, we started with the cell organelles. I think it was already 2 weeks ago, and assigned that endoplasmic reticulum, Golgi apparatus, and lysosomes are part of endomembrane system. Here we have a picture that lysosomes have their own membrane. Yes, and also inside we have these hydrolytic enzymes which are able to digest, recycle, metabolize, and degrade, break down all of the particles which are not essential anymore. Lysosomes are also part of endomembrane system. Golgi apparatus, it also has its own membrane as well as endoplasmic reticulum. The other organelles we mentioned, it was a little bit later, and we were talking then about chloroplasts and mitochondria. They have also their own membrane. They have inner membrane and outer membrane, basically two layers uh, of membranes. And that same applies for the last organelle we were dealing with, and it was nucleus. Nucleus, again, outer membrane, inner membrane. It stores mostly the genetic material. Okay, so if we get back to our question, a membrane-bound organelle could be any of those we just mentioned. Okay, the energy currency of the cell. What do you think energy currency could be as an answer?
ATP.
Perfect. Great, Tomohiro. Yes, ATP. ATP is one of the best forms how the cell is able to utilize and produce and also store the energy inside of it. So ATP would be great currency regarding energy. Great, Tomohiro. A type of protein found in cells. Okay, so we have plenty of proteins and also 20 types of amino acids. So there are that will be part of your studies in the first and second year of the medicine, so we don't have to bother us with so far with that. Nevertheless, great answers so far. Good job, guys. Okay, Risa, please, can you read?
What is DNA replication? A is the process of converting RNA into DNA. B. The process of converting DNA into proteins. C. The process of synthesizing a new DNA strand from an existing DNA template. D is the process of producing energy from glucose molecules.
Thank you very much, Risa. Okay, here are the results. Risa, what have you chosen and why?
I chose B, but it must be wrong. But I'm not sure why C is correct.
You're not sure why C should be correct. Okay, so
Yes.
you went for B, the process of converting DNA into proteins. And most of the people went for the process of synthesizing a new DNA strand from an existing DNA template. Okay, so we will differ those two processes which are here. First of all, let's describe the first one, the process of converting DNA into proteins. Guys, do you remember how is the or what processes or what steps there are from the converting the DNA information into making up proteins? What steps we mentioned? First of all, what needs to be done? If we have that DNA, what we need to synthesize firstly in order to make a copy of it and in order to create a protein as an following step. This step we call transcription. Okay. Do you remember what the transcription is, guys? How it starts and what is an outcome of transcription?
It is making RNA copy to using RNA polymerase.
Hmm. Perfect, great, Ayane, exactly. So the outcome of transcription is mRNA. If you remember, we were using this analogy that nucleus is kind of library for us, and there are plenty of books. You So you want to cook a protein, so you want to cook basically something. So you need some kind of cookbook. You get into the our library, you get into library, but this library does not allow us to get the cookbook outside from it. So what we do? We have to be sophisticated. We just copy the recipe. So basically we have a new pen and paper and we copy the recipe, which is our our cookbook is the DNA basically, okay? And DNA sequence. Only some sequence or part or genes in really medical terminology, the genes we need to copy, and our new copy is mRNA, messenger RNA. If we when we are done and we have a polymer of mRNA, we get outside from our library or outside of the nucleus, and the following step is on a stage now. How do we call this step and what is it? Do you remember what is necessary for this step? Translation. And what's going on during translation? Our polymer of messenger RNA was synthesized, yes, and it is bonded or attached to the ribosomes, where another type of RNA steps into, and basically this tRNA has those three parts we call codons or triplets, and they are complementary to our codons in the mRNA. Basically, what's happening? The tRNA molecule arrive, it lands on our mRNA polymer, and it attaches amino acid molecule to already created protein polymer or polypeptide polymer. Basically, this is our protein. So if we are talking about converting DNA into proteins, we have two steps. First one, transcription, where we transcribe DNA information into messenger RNA, and then outside from the nucleus in the cytoplasm, the messenger RNA is translated into proteins, and we have our proteins. Okay, so if we get back now, this one, the process of converting DNA into proteins would not fit to the question what is DNA replication. Okay, Risa? Understand? Perfect.
Yes.
Perfect. Okay, so the B is now clear. Do you remember how do we call process of converting RNA into DNA? It was really like additional information, but we mentioned one example if you remember. It's called reverse transcription. Reverse transcription, and we mentioned an example reverse transcription, HIV virus. Okay, is not something relevant for your stage of studies, but you will deal with these processes during your fourth year in the medicine. But it can, for you, it can be interesting how does HIV infects HIV virus infects our bodies, and it's via reverse transcription. Basically, it converts RNA into our DNAs. So when the person is infected, it's infected with the RNA nucleic acid, and then this RNA nucleic acid is converted into the person's DNA. Okay, The process of producing energy from glucose molecules. Do you remember the process when how it is called when the glucose is broken down? We invest 2 molecules of ATP and the outcome are 4. So basically the net asset is 2 molecules. Do you remember?
Glycolysis.
Perfect. Great, great, great. And I will just show you the picture. Glycolysis it is. Yes. So we have a glucose molecule at the beginning. We trigger or we are starting the pathway with investing 2 molecules of ATPs and we have 4 as an outcome. Okay, so glycolysis would be an answer for that. Sorry. So what is a DNA replication? The process of synthesizing a new DNA strand from an existing DNA template. And that's the thing we were discussing as the last process in our chapter, what's going on in the nucleus. And as you remember, we have one DNA and then we have 2 DNA molecules. Okay, what's going on? It's a semi-conservative uh, method or process, let's say, process where what does it mean basically that at the end we have one strand is the newly synthesized and the other one is the old one or the old strand. If you remember, we had leading and the lagging strands, uh, and they are a little bit different because the DNA polymerase, which is an enzyme which synthesizes new DNA, is able to synthesize DNA only in the direction from 5' to 3' basically. In the leading strands, there is no problem to do it, so we do it continuously. On the other strands, on the lagging strand, we have Okazaki fragments. Basically, what is it? Um, those are the fragments because on lagging strand it can't be synthesized continuously because we are in the opposite direction. So basically what we need always are those primers, new primers where a DNA polymerase just hops on it and synthesize till the next primer. Um, we already saw the video for this very abstract topic. Guys, if you are struggling now with this recapitulation, please just follow the video. I uploaded to the Google Classroom. It's very nicely described, it's very nicely explained. We already covered that. So if you have any questions, please ask, but we can't watch the video one more time, okay, regarding this topic because we were already there and we discussed it. Do you have any questions so far to replication or transcription or translation? Is it clear or do you have any questions? No? No questions? Can we move on? Perfect. Okay. Also, guys, please just maybe check the recordings. We were discussing it very slowly. Now we are only recapitulating the things. Okay, so recordings. If you want to recap the topic, just play them. Okay, that's the reason why we upload them. Perfect. Okay, okay, okay. No, here we are. Okay. What is cell cycle? Hitomi, please, can you read?
What is the cell cycle? A. The process of the ordered sequence of events that a cell undergoes to grow, replicate its DNA and divide into two daughter cells. E. The process of converting RNA into DNA. C. The process of converting glucose into energy. D. The process of protein synthesis.
Thank you, Hitomi. Mm-hmm. Okay, so here are the results. Hitomi, why have you chosen A?
Um, because cell cycle is process of cell grow and replicate its DNA
Mm-hmm.
and divide.
Yes, perfect, exactly. Um, Let me show you the picture. So as you remember, we divided cell cycle in basically two phases. The M phase, which is very short phase, comparing to the other phase, which is interphase. And then we divided interphase into three subphases, G1, S, and G2. And everything going on, or every cell which is in the interface and it's not in the G0 phase, okay, is preparing for M phase or cell division. We have this very simplified table which describes what's going on in the cell uh, regarding each phase or during each of the phases. G1, there is basically cell growth. During S phase or is DNA synthesis or replication. DNA replication is nothing else as we just discussed a few minutes ago. And this process, DNA replication, when we are creating from one DNA molecule the two DNA molecules in order to have enough genetic material in both cells, is happening during S phase of interphase during the cell cycle. Okay, and we are doing it as we are doing it, or DNA is replicated just because to have enough genetic material in both daughter cells. Okay, and also during a G2 phase growth and preparation for mitosis, we mentioned that uh, the organelles are synthesized uh, and also enzymes or additional proteins which are essential for new cells or for the other cells. Mhm. Okay. Okay. So how do we call the process of converting RNA into DNA? Do you remember?
Reverse transcription.
Perfect, great. Reverse transcription. HIV could be a very good example. Thank you, Tomohiro. Uh, RNA into DNA. The process of converting glucose into energy. Anyone? glucose into energy? What's the process during which one we invest two molecules ATP, and then as an outcome we have four? It's
Glycolysis.
perfect. Glycolysis. Great. And the last one, the process of protein synthesis. When the proteins are synthesized.
Translation.
Perfect. Great. Where does translation takes place? Where in the cell?
Ribosomes.
Ribosomes. Very good. Ribosomes. Where we are also able to find ribosomes? Here we are able to see that the ribosomes are just freely located, or they are just freely floating inside the cytoplasm. Okay, so they are attached to non-structure, or to any other structure. However, there is one structure which have ribosomes on its surface, or one organelle, better to say. Which organelle has plenty of ribosomes on its surface? Also, the name is or its name is derived from that the ribosomes are on it.
Rough ER.
RAF ER, yes, RAF endoplasmic reticulum. Perfect. RAF endoplasmic reticulum. As you see here, endoplasmic reticulum has a smooth type, or smooth endoplasmic reticulum, and a rough type, or rough endoplasmic reticulum. And it's the reason why we call it a RAF is because there are plenty of ribosomes. Those dots on the elongated tubes are ribosomes. Okay, so that's where the translation takes place. Very, very well. Also, you would be correct if you would say that on the nuclear envelope can be ribosomes attached. However, the let's say like number one organelle, where the ribosomes are accumulated or concentrated, is definitely RAF ER. That's great. What you explained. Perfect. Okay, okay. Where are we now? Perfect. Let's do the one more time, one more question. Ayane, please, can you read?
What is the main purpose of mitosis? A. To produce gametes, sperm and egg cell for sexual reproduction. B. To generate genetically diverse offspring. C. To repair damaged tissue and heal wounds. D. To ensure the distribution of identical genetic material to daughter cells.
Perfect. Thank you very much, Ayane. Let's have a few more seconds. Okay. Okay, so here are the results. Let's discuss them. Ayane, what have you chosen and why?
I just A, but I don't exactly know why is it correct.
Mm-hmm.
Yeah.
Okay, so you went for 8 produce gametes, sperm and egg cells for sexual reproduction. Okay, okay. Then, Mina, what about you? What have you chosen?
I chose B, but
Mm-hmm.
mitosis uh, make younger cells,
Mm-hmm. Daughter
daughter
cells.
cell,
Mm-hmm. Yeah.
daughter cell. But so the option is I don't know the name.
Okay. Yeah. Mitosis definitely creates uh daughter cells, or the result of mitosis are two identical daughter cells. But if you remember, a very important part is S phase. Why S phase? During S phase, the DNA is replicated. So basically, what is an outcome? Let me use this slide. We have one cell. Yes, we have one cell, and if the mitosis, or I will use this one. This will be better. Let's focus on this slide, mitosis. We have diploid somatic cell, okay? Each somatic cell, or every cell in our human or in our bodies, are diploid. Basically, they have 23 pairs of chromosomes, okay? Haploid cells, they have only 23 chromosomes, or one pair, um, 23 chromosomes. 2n represents in a human race 46 chromosomes, okay? Now the DNA replication takes place. So basically, we are still 46 chromosomes, but we have 92 chromatids. Do you remember this last one picture we were discussing on Friday? Here we have one pair of chromosomes, one from a mother, one from a father. If you remember the structure of chromosomes, we have a longer arm of chromatids and shorter arm of chromatids. Here is a centromere when spindle fiber attaches. Nevertheless, this is just one chromosome and it has only one chromatid. This is also just one chromosome and it has only one chromatid. We have 23 pairs of such a things. Okay, so 23 times more there is in each of the cell. 23 pairs of chromosomes and also 23 pairs of chromatids. So basically for this 46 chromosomes, 46 chromatids. I will draw that here. 46 chromosomes and also 46, I will use only C for chromatids, okay? After replication, we still have 46 chromosomes, as you see here, but each chromosome has additional centromere, chromatids, sorry, chromatid, I apologize. Additional chromatid. Those two chromatids on each side we called sister chromatids, and they have identical DNA. So gene located here is identical to gene located here. So genetical information on both chromatids are identical. So what is an outcome? 46 chromosomes, but 92 chromatids, because we doubled the amount of chromatids on each chromosome. Okay, so to generate genetically diverse offspring would not be able, because in each cell we have identical DNA. Okay, in each of the daughter cells after mitosis we have identical DNA, so the cells are identical. Okay, so here we start only with one cell, then the mitosis happens, and we have two daughter cells, but they are absolutely the same. Okay, so to generate genetically diverse offspring would not be able, as well as to produce gametes, sperm and egg cell for sexual reproduction, because for sexual reproduction we need two haploid cells, or one N cell, or 23 chromosomal cell. What are the results or the outcomes or products of meiosis? As you see, we have only one N, one C, so one N means 23 chromosomes, and one C with one chromatid. And when the sperm, which is one N, one C, got to the egg, which is also one N, one C, one N, one C, the outcome is zygote. And zygote is already 2N, or diploid and diploid cell, and then the zygote evolve, and all of the processes during pregnancy takes place, or this uh, is the topic of embryology. You will be covering embryology as a topic during your second semester in first year, and then after the childbirth, the child is already, and then growing and other processes takes place. However, this is the start. This is the start of a life, basically, of every human life, when the sperm cell um fertilize the egg. Because, and how is it possible to create a zygote? Because the sperm and as well as the egg has haploid haploid genetical information, or basically only 23 chromosomes. Okay, to repair damaged tissue and heal wounds. Um, Actually, mitosis takes place in some reparational mechanisms, but it's definitely not its main purpose. Okay, main purpose of mitosis is to distribute genetic material uh, to the other cells evenly. Okay, so to have two diploid daughter cells which are identical. This is the main purpose of mitosis. Um, okay, guys, is it clear so far? Mitosis topic and also the other things we were discussing, as well as replication, translation, transcription. We were also recapping the eukaryotic cell and also metabolic pathways. Any questions to this? No? Okay so far? Perfect. We still have 4 minutes left. However, we are done with the intro part of our session. What I would like to continue with you is meiosis, something we already started to discuss. However, we have only 4 minutes left before the possible break. Would you like to see the introduction to the meiosis as a video, or would you like to already start with the group work? So yes, I would like to see a video. No, I wouldn't. I don't want to see a video now. Okay, here are the results. So let's check the video, and we will do this video as an introduction to meiosis as an topic of today. Okay, so tools, desktop share. Is everyone able to see a screen?
Yes.
Perfect, thank you very much, Hitomi.
In this lesson, we'll explore the details of what happens during the phases of meiosis. Meiosis, sometimes called reduction division, is the type of cell division that produces gametes. By gametes, we mean sex cells, such as sperm cells in males and egg cells in females. Meiosis is broken down into two stages of cell division, called meiosis I and meiosis II. Meiosis I has 4 phases: prophase I, metaphase I, anaphase I, and telophase I. And meiosis II also has 4 phases: prophase II, metaphase II, anaphase II, and telophase II. Let's look at what happens during meiosis I. Prophase I starts with a diploid cell. Its chromatin contains 2 uncoiled, spread-out sets of chromosomes, one from each parent. After the DNA in the chromatin replicates, it condenses into the more familiar X-shaped chromosomes. The replicated DNA is the same in the identical sister chromatids of each chromosome. In a process called synapsis, each chromosome pairs up with and binds to its corresponding homologous chromosome, forming a tetrad. A tetrad is the group of 4 sister chromatids in paired homologous chromosomes. The chromosomes contain genetic information called genes. These genes were inherited from each parent. And different versions of the same gene on each chromosome are called alleles. In a process called crossing over, chromatids from each homologous chromosome exchange segments of alleles. Also called recombination, crossing over randomly happens on every chromosome, resulting in different gene combinations. This explains why every gamete is genetically different from every other gamete. Crossing over results in genetic variety in offspring. This is why children are different from their biological parents, as well as from their biological siblings. Continuing on with prophase I, the nuclear membrane disappears. The centrioles move to opposite ends of the cell, and spindle fibers fan out from them. Next, in metaphase I, the homologous chromosomes line up at the equator and attach to spindle fibers from opposite poles. During anaphase I, spindle fibers separate the homologous chromosomes in each tetrad and pull them to opposite poles of the cell. The cell enters telophase I, with one chromosome from each homologous pair at separate poles. However, each chromosome still consists of sister chromatids. Keep in mind that each chromosome's sister chromatids are no longer identical because of the allele exchange that happened during crossing over. Then, spindle fibers disappear and the nuclear membrane reforms around the chromosomes. Finally, cytokinesis occurs. Meiosis I ends with two genetically different haploid daughter cells. Each haploid cell contains only one set of chromosomes consisting of paired sister chromatids. Both cells now enter the next stage, meiosis II. However, unlike meiosis I, DNA does not replicate before meiosis II begins. Once again, in prophase II, the nuclear membrane disappears, and spindle fibers fan out from the two sets of paired centrioles. During metaphase II, the chromosomes in each cell line up at the equator and attach to spindle fibers from both poles. During anaphase II, the sister chromatids of each chromosome separate and move to opposite poles. Once the sister chromatids separate, they are called chromosomes. Finally, during telophase II, the spindle fibers disappear and nuclear membranes reform. And cytokinesis occurs in both cells. Meiosis II ends with four genetically different haploid daughter cells, each containing only one set of chromosomes. Some key points to remember about meiosis. It begins with a diploid cell. Meiosis only produces gametes. Gametes are genetically different haploid cells, sperm cells in males and eggs in females. Meiosis has two stages of cell division called meiosis I and meiosis II. During meiosis I, homologous chromosomes separate to produce two haploid cells, each containing chromosomes in the form of paired sister chromatids. In meiosis II, the sister chromatids separate in both cells, becoming individual chromosomes. Cytokinesis of these cells produces four genetically different haploid gametes. And here are some key points to remember about prophase I. The pairing of homologous chromosomes called synapsis occurs. Each pair of homologous chromosomes consisting of four chromatids is called a tetrad. During the process of crossing over, chromosomes in homologous pairs exchange segments of alleles. Crossing over results in genetic differences in gametes. All gametes produced by meiosis are haploid.
Okay, guys. Sorry, guys, my bad. Okay. Let's have a. Would you like to have a break, or would you like to continue without a break? Okay. So most of you would like to have a break, so let's have a break for 10 minutes. So it's 15:54. So at 16:04 your time, let's be back. Okay? Perfect. Hello. Okay, group. Hopefully all of you are rested. And yes, let's start with the core of our session. The core of our session would be today meiosis. Now, uh, we are changing now the the learning way. So so far we were just passively learning. Now we will try to actively learn something. Okay, so meiosis would be your task now to find out something regarding meiosis 1 and meiosis 2, how they are different, what are the outcomes of them, of those two parts of meiosis. Um, uh, are we just five? I thought we were six. Okay, okay. So I will tell you. I will now distribute you to the rooms, and you will work on those topics. So Ayane, Himari, and Mina, you will try to find out anything and research what does the meiosis one is. The same would apply for Hitomi and Tomohiro. Okay, but meiosis 2. Hitomi, Tomohiro, you have meiosis 2. One more question from me. How much time would you like to have? A is 3 minutes, B is 5 minutes, C is 7 minutes, D is 10 minutes. Okay, one more person. Okay, so it was tight, but we will have 7 minutes, okay, for a group work. Let's have then 7 minutes. Ayane, Himari, Mina, meiosis 1, Hitomi, Tomohiro, meiosis 2. Okay, okay, so, okay, now let's go to the first room. Okay, guys, I will just upload the picture very, or just give me 1 second, okay?
くそ長くなると思う。7分じゃ無理でしょ、調べるの、今思ったけ
ね、
ど。大きく分けて4つぐらいなのかな。
これ、
すみません。光が強すぎて見ることができません。
見えないかな？
あ、ちょっと。 順なんとかんとかですか？
うん。
どうもあ
ディ
りがとうございます。
フィニッションみたいなやつ。
Group, you have a picture from meiosis, okay? Here.
Yes.
Yes.
Okay.
いいですか？
何でもいいよ。
Ladies, so ladies, you have a picture from meiosis uploaded, okay? So you can use
Okay.
this one. Perfect.
Okay.
Any
Okay.
questions? No?
No.
No.
No. Okay, perfect.
ステージ、なんて調べて出てくる？
検索エンジン Yahoo ですか？
いや、Google。
あ、はい。
Yahoo で調べてたんか。ごめん。
僕がYahoo の方にGoogle の方に押してます。
長いはどこ見ても。
最初に出てくるの同じですね。ちょっと待ってくださいね。これです、これ。
メイオシス2って調べたら、
あ、はい。
ひらがなカタカナ
英語です。
じゃあ、
そうだね。
そうすれば、なんかだいたい222的ななんか、なる？
じゃあそしたら次のプロフェイス1とメタフェイス1のやつやるね。
うんうん。
で、最後の2個お願いしていい？
そうそう。
はい。
待ってね。
どっちやる？あやねちゃんはどっち？
プロフェイス1の2個目と3個目の。
1個目をやるってことね。
そうそうそう。
ごめん、なんか電波がめっちゃ悪い。
めっちゃ悪い。
だからうちがめっちゃ悪い多分。
は、プロフェイス1のとことメタフェイス1のとこ。みなちゃんはさ、その前、メイオシスの概略とインターフェースやるってこと？
そう。マヨシスがプロデュース4みたいな。
その後の2個やって、で、
その後2個。
うちが。
で、私が最後ね。
オッケー。
やばい、時間ないんだけど。
い
や、もう46秒じゃん。やばこれ終わった。
Hello everyone. So, guys, time's up. Now, um, I would like to ask a general question regarding meiosis, if you found it out or if you remember that. And the question is, Do you remember or do you understand the purpose of meiosis? Or what is it? Why something like meiosis exists? Or, okay, we can get to the purpose after we are done. After we are done the phases of meiosis one and meiosis two. So, meiosis one group. What have you found out regarding that?
Uh, Meiosis is a process where a single cell divides twice to produce four cells containing half the original amount of genetic information. Meiosis 1 can be divided into five stages: interphase, prophase, metaphase to anaphase, and telophase 1 and cokinesis 1. Interphase is the DNA in the cell is copied, resulting in two identical full set of chromatin. And during intercells in microtubules extend from these centrosomes.
Mm-hmm.
And next, in phase 1, the nuclear membrane disappears and one kinetochore forms per chromosome rather than one per chromatid, and the chromosome attached to spindle fibers begin to move. And then metaphase 1, um, bivalent, each composed of two chromosomes aligned at the metaphase plate. The orientation is random, with either parental homolog on the side. This means that there is a 50-50 chance for the daughter cells to get either the mother's or father homolog for each chromosome.
Mm-hmm. Perfect.
The stage in the first meiotic division after anaphase 1 and identified by the complete movement and separation of the paired homo- homolog- homologous
Mm-hmm.
homologous chromosomes to the opposite ends of the cells, and the single cells then pinches in the middle to form two separate daughter cells, each containing a full set of chromosomes within a nucleus. This process is known as cytokinesis.
Mm-hmm. Okay. Thank you very much, Mina. Thank you very much, Ayana. Thank you very much, Himari, for bringing this here. You have done your job very well. So, yes, I will maybe just provide you with maybe a few more additional informations. However, you explained it very nicely and very well. Okay. Um, uh, So regarding meiosis, or how to understand this process, meiosis. So the sole purpose of meiosis is the formation of gametes or sex cells we are able to see at the end of meiosis 2. Those sex cells we can assign as eggs or sperms, okay, in humans. Um, um, for example, meiosis is really just highly specific for creating the sex cells. If you have, for example, in your household, a cat or dog, which is neutered, okay, so there are no reproductive organs, or your house pet has no reproductive organs, there is no meiosis going on inside its body. Okay, so this is just to realize that where mitosis takes place and where the meiosis takes place. Okay, so meiosis is really just specific for creating and gametes or sex cells. Okay, mitosis takes place in all of the other somatic cells. Okay, so that's the first thing to understand, the differences between mitosis and meiosis. Um, uh, yes, as you very nicely said that uh, as an outcome of meiosis, is reducing the number of chromosomes. So we have just 23 chromosomes, and we create also an haploid or 1N sex cells or gametes. Okay. And as we have here, combining those two sex cells, um, we form one diploid zygote, which have, in our case, already here 2N, okay, or diploid zygote. So we started with 1N sperm and 1N egg. Then the sperm fertilize the egg and they form together an zygote, which is already 2N. Okay. Uh, so let me bring you back as we started. Um, at the beginning of meiotic division, specific cells in the ovaries or testes, so our eggs or sperms, we can call them also germ cells, um, appear outwardly no different from regular somatic cells at the beginning. So yes, they are no different from somatic cells at the beginning. And as before mitosis, let me show you the picture. As before the mitosis, even here we have our interface, what you very nicely mentioned and described during meiosis 1. And during this interface, we have G1S and G2 phases, so this is absolutely similar to mitosis. Also, our checkpoints are there. Those checkpoints we were discussing, checkpoints were in G1. There was one checkpoint, and also at G2 there was another checkpoint, okay, where the cell check itself whether everything is okay, whether DNA is replicated properly, and if there is mm, like each function works with their own purpose, whether the cell works properly. So those things are basically checked during checkpoints in G2, in G1, and then in G2. Yes. So the first meiotic division, or we can also say as meiosis 1, um, it has all the same phases as mitosis, but the most like attention, or we should highlight mainly the prophase. Why the prophase is so important? Because the thing we call crossing over takes place here. What is crossing over, ladies? Can you just remind us one more time what is crossing over? Have you found out something regarding crossing over? Nothing really? A lot? Okay, so I will try to explain it maybe. So, as we said, prophase 1 or in this phase, chromosome condensation and the breakdown of nuclear envelope occur. However, the chromosomes are not randomly aligned. They pair up with the one homologous chromosome from the mother and one from the father. So that's what you were mentioning exactly. So we have two homologous chromosomes. Please zoom into the picture. It will be better if we zoom in the if the, we zoom the picture more in, and uh, crossing over takes place where portions of non-sister chromatids of homologous chromosomes exchange segments. If you see on this picture what I just described, we see here homologous chromosomes as they are pairing, and then one sequence or one portion of non-sister chromatids of homologous chromosomes exchange segments. Let me show you the I will draw that, maybe it will be better. Here we have a chromosome of your father. Here we have a chromosome of your mother. They are homologous chromosomes. What after crossing over is happening? So we have crossing over, crossing over. This would be an outcome. Oh. They exchanged some genetic material on non-sister chromatids. Okay, so basically non-sister chromatids means that the one chromosome exchanged material from the another chromosome, but still from homologous pair. They exchanged, and this is an outcome. Why do you think something like crossing over exists? Do you have any idea? You can even just guess. Why something like crossing over has meaning in the evolution or in the nature? Just ideas, you can guess. Just guess. You don't have to know this. Just try to guess. There is no wrong guess. If you, yeah, just try, please. What comes to your mind?
Products more various genes.
Very good, very good. So basically, why something like crossing over exists? Because the products, or in our case, offspring, uh, increase the variability of them. Okay, so that was also in the video mentioned. If we have um, uh, cro if something like crossing over takes place, the variety of possibilities of new offsprings is almost infinite. Okay, if chromosomes were only randomly segregated without any crossing over, there would be around 8 million possible combinations of offspring. Okay, so even though this number is really large, uh, it would be nowadays, when we are already above 7 billions of people in population, there would be around 1,000 people similar to each of us. Okay, so there will be 1,000 people with the same chromosomal structure as for example I have or you have. So thousand thousand people just like you genetically. However, if something like crossing over takes place, there is almost zero chance that there will be somebody genetically identical to you or to anyone else on this earth. It's really almost a zero chance. Practically it's zero. Okay? So crossing over allows allows the population and also the societies the tremendous diversity among the offspring and also among the humankind and also in each other um, races of animals and also in plants where does this takes place. Okay, so that's the main reason, main purpose of crossing over, to increase diversity in offsprings. Okay, so that's why meiosis is really specific regarding crossing over and why it is very important. So please remember that crossing over takes place during prophase 1. Okay? If there is one thing you should remember from meiosis, this is it. That what is crossing over, what is its main purpose, and when it takes place. So during prophase 1. Group, is it so far understandable, the crossing over? Yes? Perfect. Okay. Then metaphase 1. So we have bivalence. So basically, our chromosomes continue to condense until they form microscopically visible structures called tetrads. So basically, 4 chromosomes. Metaphase. So basically, this is the same, very similar to mitosis again. Uh, During metaphase, we are able to see chromosomes under microscopes and chromosomes align in the equatorial plane. On this picture is already more mm, showing us the anaphase, the following phase. Um, when the spindle apparatus attaches fibers to each centromeres. Centromeres is just the nothing else as the center of the chromosomes when the spindle fiber attaches its fibers. And where is this slide? Yes. And as a result, one complete double chromatid chromosomes is pulled to each pole. Okay, so this is very important. Not just one chromatid for each chromosome, but 2 chromatid chromosomes are pulled towards new poles of a new cells. Okay, so we have a chromosome with 2 chromatids on one side and chromosome with 2 chromatids on the other side. Okay, so this is also a little bit different comparing to mitosis. Okay, nevertheless, during meiosis too, we can say it's very similar to mitosis, but this is something different. Even though we call it metaphase and anaphase, there are some differences. Chromosomes decondense during telophase, and the nuclear envelope forms around 2 new nuclei, each containing 23 double chromatid chromosomes. So here we are able to see that our chromatid chromosome structure is decondensed nuclear envelope is formed in the new cell in both new cells, and we have basically two two cells with 20 23 chromosomes with two chromatids. Okay, so this is an outcome of meiosis 1. 23 chromosomes, or 23 double chromatid chromosomes, let's say. So that was meiosis. What about meiosis 1? I apologize. Meiosis 1. What about meiosis 2? Guys, what are your findings?
Prophase of meiosis 2 starts after the formation of daughter cells in the cytokinase 1. The chromosomes begin to condense with the disappearance of nuclear membrane, the Golgi apparatus, around the year. In metaphase 2, the chromosomes are connected to the central poles by the microtubules. They also get aligned at the equator of the cell and form the plate.
In anaphase 2, the sister chromatids separate from one another and are pulled to opposite poles of the spindle due to the contraction of the spindle fibrosis. And telophase 2, the cyclomocytes begin to uncoil and become thin. They recognize into the nucleus with reappearance of the nucleosis and nuclear membranes in each pore.
Very good. Very, very good. Thank you, Hitomi. Thank you, Tomohiro. So let me just maybe again provide you with a little bit more additional information. However, it was very well handled. So meiosis 2. What we have to mention, or second meiotic division is another possible name for this process or pathway. Um, as we know already from meiosis 1, we have two um odd combinations of chromosomes, um, which are completely new, and what is very highly probable that the certain combinations of chromosomes exist for the first time in the species history. Okay, so it's very probable that each of you guys, Himari, Tomohiro, Hitomi, Ayane, Mina, all of you have so specific combinations of chromosomes that no one like you ever existed before you. Okay, so that's why you are basically unique from the other people around you, even though there are over 7 billion people. Okay, so we are still unique. Even yes, so what we have to mention regarding meiosis 2 when we are starting, many authors describes meiosis 2 as the same as the mitosis, but it's we have to understand the meaning. We already understand the purpose, and the purpose of meiosis 2 is entirely different from mitosis. Okay, even though the process appears similar, without duplicating DNA, it separates chromatids from each other, and it's resulting in a haploid set of chromosomes in both new cells. So we start with 2 cells and we are ending with 4 new cells with just haploid 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 number of chromosome. Okay, so prophase 2. Nuclear envelope breaks down and chromosomes condense. During metaphase, the metaphase align into the equatorial plane, and then during anaphase, the each chromatid is pulled apart from a chromosome. So this is the thing we were discussing in mitosis. So we have chromosomes, and then the chromatids are pulled away towards opposite poles. And during telophase, definite nuclei with haploid set of chromosomes are formed. Okay, and as a result, we have 4 haploid cells, each genetically unique, so they are different between themselves. So this is also very important to differ from mitosis, because if you remember, we were discussing the mitosis today. I think here, no, here, here will be the picture. So clear all. As we see on this picture, mitosis has 2 identical daughter cells as an outcome. However, meiosis has 4 haploid cells as an outcome, and each one is unique. Okay, so there are similarities, but is not definitely identical. So everyone is different. Okay, each of the 4 cells the result of meiosis 2 is different to the other cell. Ladies and one gentleman, is it clear so far? Yes? Okay, perfect. So that's basically mitosis and meiosis. Let's have one um, activity before we end, which is clinically orientated. And your task will be to find out how is trisomy 21 connected to our session's topic, and what do you know about the condition. Okay, so this is your task for upcoming, let's have 5 minutes in the breakouts. Okay, understand so far? I will tell you who is going to do work with whom, but the task will be the same. Okay, so we will do a little, we will mix you a little, just slightly, and yes, no, like this. Okay, so let's have 5 minutes for the activity. So Ayane works with Mina, and Himari, Hitomi, and Tomohiro, you work together in the other group, okay? But the task is the same for each of you, okay? Trisomy 21, find the connection to our today's main topic, and what other things you are able to find of trisomy 21. We have another specific name which is more known in the public, uh, this condition's name. Okay, so let's start. Okay, guys, so what are your findings? Do you hear me?
Yes.
Okay, perfect, because yes, perfect. So what are your findings? What is trisomy 21? I think you are familiar with the condition. At least you already heard about that.
Trisomy 21 is also called Down syndrome, and it is a genetic condition where people are born with an extra chromosome. Most people have 23 pairs of chromosomes with each cell in their body, for a total of 46. A person diagnosed with trisomy 21 has an extra copy of chromosome 21. Each means their cells contain 47 total chromosomes instead of 46. This changes the way their brain and body develop.
Perfect. Very well, Ayane.
Um, so I talk about trisomy 21 future. So and children and adults with trisomy 21 have distinct personality features. Not everyone with trisomy 21 has the same feature, but some of the more common features are flat face, small head, short neck, pro- protruding tongues, and abnormal shape or small
Thank you,
ears.
Mina. Exactly. Perfect. So here we have the features Mina was just explaining us. And Down syndrome is another name for trisomy 21. It's one of the most usual or most common genetical disorders. And here on the left side, if you zoom in a little bit more, we see the pathophysiology behind. Basically, there is a non-disjunction during meiosis 1. So basically, the chromosomes or three copies of chromosomes remain. um just in one cell, so there is no disjunction of them. And as an outcome, we have 47 chromosomes in each cell. This can happen during meiosis 1 and during meiosis 2 as well. However, 95% or in 95% of cases, it happens during meiosis 1 already. Okay, so you see this here, the empty bubbles. So basically, it refers that there is no genetic material basically. Okay, perfect, guys. Can I ask you maybe about your feedback or feelings basically? How did you like this session that we started with intro? We recapped that, then we moved to the core. You had some active learning activities focused firstly theoretically on meiosis, and then at the end we had a little clinical case activity which connected the meiosis with some real clinical case scenario. Did you like it, or would you like to change something, or how are your feelings? If you can click on that. Hmm? Okay, thank you very much. So this is a feedback. So I then uh, yes, think you like the structure basically. So we will try, or I will try to continue to structurize the following sections in a let's say similar way. Thank you very much for your active preparedness again, that you were active as well during the session. And yes, thank you very much also that you are attending the classes and that you are, would say, very hardly working students, I think, because we so far covered a lot of topics and you are dealing with them very well. So just keep this track, and I really hope you will be futures in the future very good doctors. However, we still have plenty of things to cover together. So see you tomorrow. Okay, guys. Thank you very much, and see you tomorrow.
Thank you.
Thank you.
Thank you.
Okay. So stop the recording.